I'm so delighted today to welcome Dr. Tamar Gur to the Women's Health Seminar Series. I'm very excited to listen to how microbes and microglia are connected. And I want to, before we begin, acknowledge that we live, work, and play and participate in a community on the unceded, ceded, and traditional territories of the 203 First Nations, along with the 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia. And I recognize that many of you are not in British Columbia at the moment. That's just where the Women's Health Research Cluster resides. So for all of you, please just take a moment to think about the lands on which you live. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety, and equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. And as I said, today I'm joining you from North Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of Musqueam, Squamish, and Tsleil-Waututh First Nations. And just a tiny bit about the Women's Health Research Cluster. We have over actually 150 members right now, and it's worldwide, not limited to British Columbia. That's why we should alter that a land acknowledgement a little bit. We have a lot of outreach opportunities, so podcasts, blogs, if you feel like doing some scientific communication. We have a number of events, as you know, that we run. And please join us if you haven't joined us. And we rely on support from people like yourselves, as well as University of British Columbia gives us a little bit too. So without further ado, I'm going to stop sharing my screen so that Dr. Tamar Gur can show her screen. And while she's doing that, I'm going to thank you, Tamar, for putting in your line acknowledgement. And I encourage any of you to do that in the chat as well. While she's sharing her screen, I'm going to read out her bio. So Dr. Gur received both her MD and PhD, which just blows my mind when people do an MD and a PhD at the University of Pennsylvania, where she studied the effects of selected serotonin reuptake inhibitors on neurogenesis in the laboratory of Dr. Julie Blendy. She completed her clinical training in the research track of the Psychiatry Residency program at the Hospital of the University of Pennsylvania, where she honed her research interests in women's mental health. Her clinical work at Ohio State focuses on perinatal and postnatal depression and anxiety, which dovetails with her interest on the effect of prenatal stress on neurodevelopment. And she uses a variety of approaches, which we'll hear about. She enjoys, in addition to her clinical work and research efforts, Dr. Gur enjoys working with the next generation of physician scientists in a role as Associate Director and Co-Investigator of the NIH-funded MSTP program. The NIH really likes their acronyms. So I don't know what that stands for, but she also is passionate about improving pathways for historically excluded individuals and is able to facilitate that through a role as Faculty Director in the College of Medicine Aspire program. So I just also want to say that I met Tamar a couple of times, I think, at least one time anyway, and she is actually an amazing mentor to everyone. She gives this fantastic opportunity, which I won't describe, but she is really literally a superwoman. So I know she enjoys putting gifs up on Twitter of Wonder Woman, which I loved the first film. The first film.
I did not love the second.
I did not love the second. I wanted to love it very much.
I did too.
Anyway, so at some point, I'm sure she'll share some of that with her, but I will also say that her fun fact, because as many of you know, I like to give a fun fact, that Dr. Gur's fourth word was proposal. So there is definitely an interesting story.
I pronounced it proposal.
Whatever, fourth word, that's pretty amazing. So without further ado, I'm going to give you the floor.
Yes, well, thank you so much for the lovely introduction. I was lucky enough to meet you at a very pivotal part of my career where I was really sort of going through some self-doubts and like, what am I doing? And you really made me feel like I was on the right track. So I really appreciated that. It was a very influential meeting for me when you came here to Ohio State. So, and you've given me so many wonderful opportunities since then. So I really appreciate your mentorship. So I'd start there. For any of you confused, my fourth word was proposal. My first word was ima, which means mom in Hebrew. My second word was abba, which is dad. My third word was or, which is light in Hebrew. And then they were putting in a huge grant at the time that I was acquiring language. So my fourth word was proposal. My parents, I am very privileged that my parents are very wonderful people, and also that they are scientists. My mother is a physician scientist. So I never questioned whether or not it was possible to do this and have children. I knew that it was, but I realized that it's a tremendous privilege, and that's one of the major reasons why I work so hard right now to help those who have been historically excluded access this type of incredible career as well. So today I will be speaking wearing, I think, all of my hats. There's some of all of my different hats in this talk today. There's even a picture of my kids at the end, spoiler alert. And I will be discussing prenatal stress in the developing brain, from maternal microbes to offspring microglia. And I know that Zoom is not the most interactive medium, but I did on purpose because they asked me to, but also because Zoom is not very interactive, leave time at the end. I deleted several slides to make sure that we'd have the opportunity to discuss any part of my talk today, or any questions for trainees on the call, if they have questions about career path, anything the next hours, or however long we have, the next 52 minutes are yours in Canada. So I wanted to make sure we had time at the end. My affiliations reflect my research interests. So I'm part of the Institute for Behavioral Medicine research here. It's really a powerhouse group of psychoneuroimmunology and endocrinology researchers here at Ohio State that I'm lucky to collaborate with. I'm physically surrounded by just some really wonderful experts in a variety of P&I topics. So I am a psychiatrist by training, and I'm also have appointments in neuroscience and OBGYN, which reflect my clinical interest in perinatal psychiatry. I don't have any disclosures, fortunately and unfortunately. So one of the main reasons I'm here is because I know that there's a passion in your center for women's mental health. And I'm specifically interested in maternal mental health. Of course, there's many aspects of women's mental health that lies outside of the perinatal time period. But I've really focused on this now as my major area of clinical as well as research interest. I have a suspicion that as I go through the next, I'm done having children, and I have a suspicion that as I move through different phases of women's health, I might become interested in menopause and some other things, especially cognition. But I'm just, for right now, this is where I live, in preconception, pregnancy, and the postpartum. And why do I do that? I do that because depression and anxiety are prevalent during pregnancy. There was a myth taught decades ago now that pregnancy was a protected time period. I suspect they thought we floated around on a cloud of pink pheromones, just feeling blessed to be with child. But that is far from the case. And in fact, depending on the study that you look at, between 6 to 23% of women report symptoms or meet criteria for major depressive disorder or a major anxiety disorder during pregnancy. So far from being protected, it's actually about a quarter of women will at some point in their lifetime have depression or anxiety. And so the pregnancy is not any different than that. And really, psychiatry is sort of late to the game. Psychology came to it much sooner, in recognizing that many of our disorders have an inflammatory component. And so I actually was at Dr. Glia will appreciate the fact that I was presenting at a national conference of psychiatrists, and one raised their hand and said, after my talk, and said, well, it almost feels like there's a field of psychiatric immunology. And I said, I'm going to stop you there. There's actually been a field of psychoneuroimmunology for decades, and you'll have to just join us at the table. You can't create your own table here. So, but there is increasing understanding that psychiatric disorders such as depression and anxiety do have an inflammatory component. And I really think of inflammation as being, if you look at this Venn diagram, where psychiatric disorders and stress, which is a known trigger for psychiatric disorders, where they meet. And so this cartoon is actually a depiction of me not that long ago. My youngest is now 3, so not an infant clutching my head, but certainly reflective of my time during the global pandemic. And so stress is omnipresent, especially nowadays. And stress during pregnancy is also omnipresent. So I'm really interested in understanding how stress during pregnancy contributes to psychiatric disorders, both in the mother as well as increased risk of psychiatric disorders in the next generation. And really the bulk of the research into this topic until now has focused on a few key areas. And so just to sort of know we knew before I launched my lab, a lot of the work, and you'll have to forgive me, some of the Zoom boxes are right over my words. So I apologize if I'm saying redundant things. It's like I can't quite see all my words, but inflammation, the HPA axis, and epigenetics were really the key areas of focus for scientists in terms of thinking about how does stress during pregnancy contribute to increased risk of psychiatric disorders. Now, there's a preponderance of epidemiologic, clinical, and preclinical work that suggests that there's a critical window during pregnancy during which, if there is a stressor, the offspring that have been exposed to stress during gestation have an increased risk of developing autism spectrum disorder or schizophrenia, depression, and anxiety. And some of the finest work in this area is actually from Canada. You guys, I've had these ice storms described to me countless times, and they sound horrific, but there was, after those ice storms in Canada where people were trapped in their homes for weeks on end, women who were pregnant during that time period had an increased risk, their infants had increased risk when they go into childhood and adulthood of having psychiatric disorders. So that was really one of the formative studies that I remember reading about early on in my interest in this field. So the way this was thought to be transmitted to the next generation in terms of inflammation, some of the work, especially for maternal immune activation models, which I'll spend some time on later, was the idea being that maternal immune system is activated and cytokines, which is the messenger of the immune system, is capable of crossing through the placenta or being generated by the placenta and enter the fetal circulation, where they're thought to affect the developing brain. The HPA axis, work from your countryman, Tim Oberlander, and others have shown that there's increased placental CRH that can influence the developing fetal HPA axis. So infants born to mothers who were undergoing high levels of stress during pregnancy had a more reactive HPA axis. So there's dysregulation of the HPA axis response in the next generation. And there's some preclinical work that dovetails nicely with that work. Epigenetics. So not only do you get genes from your parents, the way that they are expressed is regulated, epigenetic regulation. So methylation, and then there's other forms of epigenetic regulation. So increased methylation of important genes in a variety of psychiatric disorders are modified by stress. So those are the three major areas of research when we think about stress during pregnancy. And the microbiome, when I was starting my research program, had just sort of entered the fray. John Cryan, I think of as the king of this topic, had written a really interesting review paper on the gut-brain axis. And I remember thinking, well, could this be important in pregnancy? And that was really what launched my research program. I was reading papers at a conference, and that there was nothing more than that. And then I Googled, this is for the trainees, I literally Googled OSU stress microbiome. And that's the way I found the best mentor I've ever had in Mike Bailey. So really Google stuff if you're interested in. Sometimes it will pay off and launch your research career. So that's where my research really has focused over the last several years now. So the first time I gave this talk, a psychiatrist came up to me afterwards and said, the brain's all the way up here and the microbes are all the way down here, and sort of gestured awkwardly at his abdomen and his butt. How do the two converge? So I went ahead on my flight home from that conference, and I made this slide because I figured if he was wondering that, there probably is more than one person in the audience also wondering about that. So stress, I think we'll all agree, impacts the brain. We've all had trouble finding we've all lived through this global pandemic. I used to have to think of examples. Now I can just say stress, and I think we all sort of know what we're talking about. But everything ranging from daily hassles to global pandemics, and now very sadly war conflicts across our world are stressful, and I think we can all have experienced that it affects our thinking, affects our mood, and so it's not too much of a stress to understand that it's affecting the brain. Well, and I'm going to focus on pregnancy for the purpose of this slide. That can lead to a myriad of effects, one of which is the activation of the hypothalamic pituitary adrenal axis, or the HPA axis for short, which I briefly introduced in our last slide. That results in the release of ACTH and glucocorticoids from the adrenals, as well as activation of the sympathetic nervous system. You might have learned about that as an undergrad as the fight or flight response. And then in this graphic, the individual is pregnant. Both the release of glucocorticoids as well as the activation of the sympathetic nervous system. I think we've all heard that saying. I was so scared I almost pooped my pants. I did not use the S word, Dr. Glia, though I do understand that that has previously been recorded in this speaker series, can lead to activation of the intestinal epithelium and the movement of feces through the intestinal tract. And so that can clearly, we now have a connection between your brain and your gut. But more than that, both the sympathetic nervous system as well as HPA axis can regulate our immune system, depicted here, as well as the release of cytokines. And cytokines themselves can also work on the microbes that are present in your tract. Now that I haven't formally introduced the microbiome, I think I took that slide out, and my apologies, but the microbiome, don't get scared. I know it can be, if that's not your area of interest, it can be intimidating. I remember feeling that way, but it's no more than no less than all the microorganisms that occupy every orifice and every surface of your body. And every orifice and every surface has its own unique signature of microbes that are present there. And collectively, the genes in those microbes are referred to as the microbiome. So your oral cavity has a microbiome, your gut has a microbiome, and they're distinct. And they're not just quietly along for the ride, they're actually producing a lot of very important factors in your health as well as your mental health. And so for the idea being that stress then impacts the community of microbes that are in your gut and that are an active participant in your health, and stress might have a negative effect on the microbes that are living and working in your gut. And so specifically, what I'm interested in is a few things. One is that stress, work from my mentor's lab showed that stress increases the translocation of microbes into internal organs such as the spleen. I was interested if that was happening. The uterus is an internal organ. Is it affecting that at all? The immune system, microbes, again, are microbes. What does your immune system do? It does a variety of things, but one of the things it does is deal with microbes. So is it altering the maternal immune landscape? And what about the fetal immune landscape? At delivery, microbes are seeded into the infant, and I'll talk more about that shortly, through either C-section or through passage through the vaginal canal. And that actually has been shown to have remarkable effects on infants' gut health and other health outcomes such as asthma or asthma and eczema, things of that nature. There seems to be a difference in children who are born through C-section. And then finally, these are three little molecules here that are active metabolites of the gut, and they're three of my favorite. The bottom one is serotonin, tied with oxytocin maybe for my favorite signaling molecule. And the short chain fatty acid is right there in the middle. And so, you know, short chain fatty acids are involved in epigenetic regulation. And so the gut is actively making metabolites that participate in our health. And so they're not just quiescently sitting there living their best life, they're actually producing metabolites that are capable of influencing our health. So the hypothesis that I'm setting out to test here is that stress-induced alterations in the maternal microbes, which are again all the way down there during pregnancy, alter the intrauterine environment, neurodevelopment, and the microbiome in the offspring in a way that increases risk of developing psychiatric disorders. And I illustrated this here with a picture of my daughter who's now 7. And in she was just such a happy baby. The lower picture is actually about as angry as she got. And I'm happy to report now, this is a longitudinal study, she's still about one of the happiest people I've ever met, and is a joy to be around. She's also a ginger, which is fun for all of us. But that's the hypothesis that I'm testing in my research program, and that I'll be discussing with you today. So we do that largely in a animal model of prenatal stress that we have recently bridged out into a clinical model. And I'm not going to be sharing results with you today from that. You'll have to invite me back, but they're very exciting, and I look forward to publishing them hopefully in the nearer future. But we did start a clinical study right before COVID, got interrupted by COVID, and now we completed it during COVID. And I think that our findings in our rodent model will be recapitulated in humans. So that's stay tuned for more about that. But for right now, for rodents, what we do in our stressor is between gestational day 10 and gestational day 16, which, as I've noted here, is this roughly corresponds to the second trimester in humans. Though you will note that in a rodent, rodents do deliver in approximately what corresponds to the third trimester in humans. So first of all, mice are not people. We shouldn't anthropomorphize any of these findings. You'll only ever catch me saying anxiety-like behavior. But I think this is a major caveat for this model, is that a large amount of their neurodevelopment does occur outside of gestation. So that's something to be noted right up front. So between embryonic day 10 and 16, or gestational day 10 and 16, they undergo a restraint stress between the hours of 9 and 11 a.m. I should tell you, this is probably a pretty mild to moderate stressor. I have colleagues who do 12 to 14 hours of restraint, but I was concerned about abortion and causing physical trauma to the mice. And so this is the paradigm that we have adopted. This is notable also because there are several key developmental milestones between gestational day 10 and 16, including placental circulation comes on board at day 10.5. So that's when the placental circulation starts. And it's also when microglia, right before then is when microglia come out of the yolk sac and start, microglia are the innate immune cells of the brain, so the immune cells that reside in the brain. They come on out and travel from the fetal yolk sac into the brain at about day 9.5. And really day 10 and 11 is when a lot of microglia activity entering the brain and dividing starts to happen. So those are important time points that I'm interested in neurodevelopment. One cohort is sacrificed at gestational day 17. Other cohorts are allowed to go through parturition and grow into adulthood when behavioral testing is performed. I want to point out, actually, this is another one of my daughters in this picture. I gave a picture of her as a baby to the artist when he was coming up with this figure for our review article and trends. But so every time I see this slide, I smile because it's like little baby her in this uterus. There are a lot of other differences between mouse and human pregnancies, one of which is that the human uterus usually houses one, but of course can house two or three, or thanks to reality TV, we know like 8 babies, but it is very different than the rodent uterus, which is bicornate. So it's more like the rodent uterus is more like a pearl necklace, with each fetus individually housed in its own uterus with amniotic fluid and its own placenta. And in our experiment, we actually tease them all out and are able to tell the sex of the fetus. Of course, we double check by genotyping it, but we're able to tell the sex of the fetus and the sex of the placenta. So therefore the sex of the placenta, and collect amniotic fluid. And I'll be showing you later on a surgery that we do. And so I just wanted you to sort of have a mental image of what I'm talking about when I talk about placenta, when I talk about fetal brain. We're able to dissect that out. We're able to dissect out fetal liver and a variety of other tissues as well. If we were in person, I would ask if there's any questions before I move on, but Zoom makes it hard, so I'm just going to keep going. So when we looked at the behavior in the moms, we saw an increase in anxiety-like behavior in the moms, as evidenced by the fact that they had a reduction in the time spent in the open arm of an elevated plus maze. So mice, except for mice in Philadelphia, prefer the dark. Mice in Philadelphia seem to not have a preference for light or dark. They would scatter across my kitchen when I lived there, regardless of if it was dark or light. But in general, mice prefer the dark. And so if they are spending a reduced amount of time in the open arm, which is lighted, that is reflective of anxiety-like behavior. So our stressor was sufficient to elicit this in the moms. We then, this is work from my talented postdoc, Adrienne, who is now an assistant professor with her own research program at the University of Illinois. She was interested in characterizing the dam, which is the correct term for a mouse mom. Though we did call her paper the dam paper for a while until it started taking shape. So this is, in this slide, I'll be talking about how prenatal stress activates the glucocorticoid pathway and induces central gene expression profile, which is consistent with depression and anxiety-like phenotype. So what I'm showing you here on the left-hand side of this figure is that there was a significant increase in serum cort in the dams when compared to non-stressed counterparts. And again, here's a nice illustration of the HPA axis. We then looked at CRH, corticotropin releasing hormone, in the hypothalamus, of course, a brain region, very important. in neuroendocrinology, and we found a significant increase in that hormone, which dovetails nicely with the serum court. There was a trend towards an increase in the BDNF in the amygdala, which has been found before in rodent models, where there's increased anxiety-like behavior. And we also saw a significant reduction in the hippocampus of TPH2. For those of you who don't have your serotonin pathways memorized, serotonin, this actually reminds me that I forgot to introduce tryptophan in that previous slide, a few slides back, about all the favorite things that poop makes. One of them is tryptophan. So I'm sorry, I skipped that one over. I see it now here again, and I realize I left it out. I talked about short-chain fatty acids, and I talked about serotonin, but I didn't talk about tryptophan. Actually, that is a major source of, your diet is the only source of tryptophan, and your microbes are responsible for metabolizing it. And so a significant reduction in TPH2 is found in the moms, which for us confirms the deleterious effects of this paradigm, so the anxiety-like behavior, as well as these changes. So we did something to them. We did something to moms. We put them in tubes for a few hours a day. And so then again, I started off by saying I was interested in what impact this has on the next generation. So this is work that we first published in Behavioral Brain Research, and then Helen Chen, the talented student who's about to defend in just a few short weeks, whose work I will be showing the bulk of today. The bulk of the data I'll be showing today will be from her. Did a 3-chamber social behavior test and recapitulated the findings in her translational psychiatry paper. I'm just going to start this video. And what you see here is 3 chambers. This is the mouse, the offspring that's either been exposed to stress or is a control. I don't remember. In one of these chambers, there's a conspecific mouse, so same sex, same age, but of a docile mouse line that is known to be very friendly. And in the other little cage is an object, it's a paperweight. And so what we do is we measure the amount of time the mouse prefers to socialize with a novel mouse versus the amount of time it would prefer just to be with the object. And what we found, and we've recapitulated this so many times now that even I believe it. For the PIs on this talk, you know that it's just reproducing your data. There's nothing quite like it. That there is a reduction in social behaviors following our prenatal stress paradigm. So this is a heat map, a representative heat maps. And then this is the graph that over the different, our sample size was 11 to 12 in this. So there's a reduction in social behavior in the offspring in the next generation. So we are doing something to the moms. It's resulting in reduced social behaviors in the next. So really it's this question of how are these peripheral changes in maternal microbes transduced to the developing brain? How is stress to the mother being transmitted to the next generation? And so we again are really interested in the microbes. So could there be increased translocation of oral and gut microbes or up through the vaginal canal? Is it inflammation? Is that playing a major role? And what the different neurodevelopmental outcomes that have been cited in the literature include changes in cognitive function, increased anxiety-like behaviors, increase in neuroinflammation, as well as a decrease in neuromotor function. So at the beginning of my talk, I'll start, not in the beginning, I think I'm already in my middle. The next thing I'll talk about is vertical transmission, which is, I mentioned in that original slide, seeding at delivery of microbes from the mother to the offspring. So ecological succession is this concept that at birth, again, through either passage through the vaginal canal or, at C-section, infants are seeded with mostly skin microbiota, is dysbiosis, or the negative changes I've discussed with stress of the microbiome in the mother. Do they alter the microbiome in that next generation? Because if you are seeded with microbe A, then that's most likely to then lead to microbe B and microbe C. If you're seeded with microbe X, then microbe Y and then microbe Z are the most likely to take root and grow and flourish. And so that could be, microbe Z and microbe C could have very different outcomes, health outcomes and mental health outcomes. So that's that principle. And so the first thing I need to show you is that when we looked at the microbiome analysis, and so this is now the maternal microbes, And this is a PCOA plot, and it's not showing you anything more complicated than each of these dots represents all the genome, the entire microbiome of one mouse. So each dot is one mouse, one mom, one dam. And how the more similar one individual's genomes are to another, the closer together they'll be, and the more dissimilar, the further apart they'll be. And here blue is not stressed and red is stressed. And what you'll see is that there's a significant difference in the microbiomes of the female mice that were exposed to stress when compared to the moms who were not exposed to stress. So yes, there was a shift in our moms with stress, and we've now recapitulated this. It feels like countless times that stress in our model leads to a shift in the microbes found. We were very interested in the significant reduction in Parasittarella, which is a very key microbe in maintaining the host's immune system. It has a very important effect on host microbe interactions and gut health during pregnancy in a clinical study. So we found a significant reduction in Parasittarella in our model. And actually a very active research project in lab right now is following up on that and actually giving Parasittarella back and seeing if we can mitigate some of the negative effects of stress to the offspring by replenishing this important microbe. So that was one important finding. And it also alters the microbiome in the offspring. So again, this is a PCOA plot. Blue is no stress, red is stress, and what you find is a significant difference between the female offspring. Again, they were perturbed. They were only perturbed during pregnancy. There wasn't a second hit or anything, but that was sufficient to really shape their microbiome into adulthood. And what you're seeing here on the right is the relative abundances of Rickenellacea, which is another microbe that's important in host health, significantly correlated between the moms and their female daughters. So it really does speak to this concept of ecological succession, but also of the relationship between the mom and the daughter, not just through emails and text, but through their microbes. And I think that that's really, really interesting and speaks to the hypothesis. So vertical transmission seems to be one potential way through which changes in maternal microbes is transmitted to that next generation. I didn't show the male microbes, but there was also a significant shift in the male offspring in their microbiota, in their microbiomes. We next turn our attention to the immune system, which quite honestly is what I think about most now, is how is stress affecting the maternal immune system as well as the offspring immune system. So we're going to turn our attention to that now. So this is a figure from a dear friend of mine, Lisa Hansu, another Lisa Hansu. Sorry, Lisa, I really think of her as Lisa. And the question there being is that there's this really complex interplay between, I think already the HPA axis in pregnancy is awfully complicated. It's wonderful, but awfully complicated because pregnancy is really a long period of immunosuppression. It had to be because you're growing an alien in your body, bookended at both ends, both at conception and parturition by inflammation. And so, and as I mentioned very early on in my talk, you know, the HPA axis is activated by stress or dysregulated by stress. I have to choose my words so carefully because I'm getting you data every day that's making this picture even murkier. But the immune system can work on the immune system to cause immunosuppression. And then there's prenatal programming that occurs of the offspring's immune system as well. And so there's important signaling during that time that looks like it can shape the immune cells in the offspring, and then they go ahead and live their lives that could make them more susceptible to psychiatric disorders, autoimmune disorders, a variety of disorders we know the immune system can play a part in. So this was my original hypothesis. I really thought, I think I got a grant to support this hypothesis, that the mother was like a faucet and turned on her immune system, and the baby's brain was like a sink. I'm sort of blurry in my background, so my hands, I'm not sure you're really seeing this. The baby's brain was like a sink, and you turned on the mom's faucet, and the inflammation, cytokines were pouring into the baby's brain and causing neuroinflammation and changing the microglia, those immune cells. And so that's what I thought. And that makes sense based on some of this preclinical literature and some of the clinical literature. So maternal immune activation is a very commonly used model where you give either LPS or poly-IC during pregnancy, and that incurs the wrath of the immune system. IL-6, everyone's favorite cytokine, shoots up. IL-1 beta, everyone's second favorite cytokine, shoots up. TNF alpha can go up, and robustly in the maternal plasma, in the fetal brain. And so that's really, I think, what I was basing that hypothesis on. And then, you know, in human studies, women who have the flu during pregnancy have an increased risk of schizophrenia. And we all know the flu sucks and makes you feel very sick and causes robust inflammation. So I really thought that this is what we would find. And so I don't know if you can already tell that that's not what we found. But yeah, that was my original hypothesis. I'll just be upfront with that. And for any trainees there on the call, or any of us, any PIs on the call who have loved a hypothesis so much, only to let it go, you know, I just want to say I'm right there with you, because this was my hypothesis. So during pregnancy, inflammation was transmitted on almost like a 1-to-1 ratio, poured into the baby's brain. And so what I really thought was that the shift in microbes was leading to an increase in CCL2. CCL2 is a chemokine that recruits immune cells to the site of injury, be that the gut, be that the brain. So my colleague John Godbout does a traumatic brain injury. CCL2 is really important in recruiting microglia to the brain in that model. There's been a lot of really elegant work done showing that CCL2 recruits immune cells to the site of injury through release of cytokine, and then they produce cytokines like IL-beta, IL-6. Epithelial cells are also capable, which I've illustrated here, are also capable of making cytokines. Cytokines and CCL2 can really come from, you know, anywhere. Epithelial cells, endothelial cells, as well as immune cells. So I thought this because I found a significant increase. Helen, found a significant increase. How about that? We found a significant increase in the placenta. This is now protein on a protein level of CCL2. So we dissect the mice I showed you earlier. In the amniotic fluid, there was just a ton of CCL2 in the placenta. There was a significant increase of CCL2 in stress. So I really thought and proposed that we would just see this influx of immune cells into the intrauterine environment, and they were going to just pump out these cytokines that were going to pour into the fetal brain. Well, we're going to revisit the elegant work of Adrienne and what she found. In fact, when we look at prenatal stress and the maternal response, as I mentioned, CORT goes up, CRH goes up in the hypothalamus, the glucocorticoid receptor, and our 3C1 looked like it increased or trended towards an increase in the amygdala. But, you know, CORT's really immunosuppressive. And this was sort of my first clue that maybe I was off. When we looked at the spleen, which is a very important immune organ, we actually found a reduction in spleen weight reflective of immunosuppression, and a reduction in cytokine production, illustrated here as IL-1 beta. So then it was like, well, I had to remind I'm married to an obstetrician. He again likes to remind me that pregnancy is an immunosuppressed state. So then we looked at the placenta immune cells. And there wasn't, even though there was this increase in CCL2, there was not a huge influx of immune cells to the placenta. And I've illustrated this in believe me, we looked in every possible way. So here on the left, we have immunohistochemistry and a representative bar graph of CD45, which would label all immune cells. We actually found a significant reduction in the immune cells in the placenta. And then we did flow cytometry, looking at CD45 positive, CD11B positive cells, and found a significant reduction there. So I was glad the data agreed with each other, but I was just shocked. I can't tell you how long I stared at these figures after Adrienne left my office that day. So there was not, even though there was an increase in CCL2, there was not this influx of immune cells to the placenta. In fact, there was a significant decrease. We looked at the fetus. We found a significant increase in CCL2 in the fetal liver. So this is gene expression. We've now recapitulated this with protein level as well. And there's also a significant increase in the fetal liver of everyone's favorite cytokine, IL-6. And there's a robust increase in microglia as well as IL-6 and CCL2 expression in both in the fetal brain. And we've now replicated this a few times. So what is that telling me? The fetus is where the signal is coming from. So prenatal stress leads to increased cytokines in the fetal and adult brain. So then we looked out into adulthood and found that so on the left here is fetal brain gene expression. Again, there's an increase in IL-6, TNF alpha. And then on the right here, we have IBA1 staining. So IBA1 labels microglia. There's representative microglia in this figure here. So we look at the motor cortex, which was not a brain area that I would hypothesize would be affected by stress. And in fact, there is no increase in IBA1 labeling in the motor cortex, but in the prefrontal cortex, and a brain region that is in fact very important in anxiety and mood and social behaviors, we found a robust increase in IBA1 labeling. There's also in adulthood increased cortical levels of IL-1 beta as well as IL-6. So this neuroinflammation is there in the fetal liver, in the fetal brain, in the fetal, and I don't think I've shown you yet, but in microglia labeling in the fetal brain as well, and then into adulthood. So there's robust neuroinflammation and liver inflammation in the fetus. So then we ask the question, is this increase in cytokines microbe dependent? And there's a few ways to look at microbes. They're all flawed, so you should do both, is my short answer, in case anyone asks me. But on the left here is germ-free mice. Germ-free mice have been raised over 10 generations in a sterile environment, and there's no microbes, viruses, fungi in them. It's very hard to do, and they're very expensive, and behavioral testing is a nightmare. And they're also very weird mice. There's now a preponderance of evidence from John Krein's group and others that show that germ-free mice have aberrant behaviors at baseline, and they have aberrant neurodevelopment, and their aberrant immune systems, and everything is aberrant, just aberrant. And the other way to look at microbes is to deplete them with antibiotics. So you deplete the dam with antibiotics, and then you can do the stressor and look that way. So we did that. And what we found here on the top, I'm showing antibiotic treatment. On the bottom, I'm showing germ-free mice. And in both, you can see that stress in our model does not increase expression of CCL2 or IL-6. And in the bottom here, we also have TNF alpha. So there is no increase in neuroinflammation in the absence of microbes. And we now also have data from the germ-free mice into adulthood and replicate that finding. Is the increase in cytokine CCL2 dependent? CCL2 is proved to be incredibly complex. And so we took CCL2 knockout mice, and so they don't have at baseline any CCL2 in any of their tissues. And we went ahead and looked into in both adulthood as well as in the fetal time points, and looked at neuroinflammation. And what we found is that no, CCL2 mice do not have increases in IL-6 or TNF alpha. If anything, they have a significant reduction in IL-6. And what really excited us was that in the absence of CCL2, there is no reduction in social behavior. The offsprings are spared the reduction in social behavior that was seen in wild type mice. And this paper came out in Translational Psychiatry at some point during the pandemic. Some time is lost meeting. And this is really exciting data that Helen has been putting together. So she's defending in a few weeks. She went ahead and she's preparing this manuscript right now. It should say Chen et al. In preparation down there. She's actually probably working on that at this very moment. She went ahead and did this really cool experiment. I thought about showing you the movie of her doing the injections, but I didn't know if some of you were squeamish. So I'm not showing you the movie. Here's just a cartoon where she did intra-amniotic injections of recombinant CCL2. And what she found is that it specifically increased the CCL2 you see here in the amniotic sac. And nothing happened in the mom's maternal plasma, and in the placenta, there was no increase. So it really did target it towards the fetus. It increased CCL2 in the plasma, and it increased CCL2 in the liver, just like stress does. And it increased CCL2 in the fetal brain. And I didn't put it on the slide because she's analyzing the second cohort and she wouldn't let me. But in the first cohort, and I don't have the reproduced the second cohort yet, but in the first cohort, there was a significant reduction in social behavior in those offspring. So it looks like we're recapitulating the effect on behavior as well. So this was really exciting for her, and I'm glad for her that this experiment worked. The last experiment of your PhD never worked, so I'm actually very pleasantly surprised right now. And so CCL2, as I mentioned, I think microglia are really important. So I didn't realize how long I would explain things. I'm a little bit short on time. So I'm going to try to move through this in a more direct fashion. So microglia, again, are the innate immune cells of the brain and likely play an important role in neurodevelopment. And that's because they have so many important tasks that they do. You know, they make cytokines, they guide axons, they phagocytose synapses, and a variety of important things. And so they really are at that a lot of important roles that I think could be playing into reduce social behavior. So we went ahead and took a look at the fetal microglia. And the first thing we found, poor Helen Chen is preparing a lot of manuscripts, was that there is a significant increase in the fetal microglia at embryonic day 17 and a half, as evidenced by IBA1 staining. So following stress, so stress does increase the number of fetal microglia. We also, Helen is very talented and managed to isolate microglia using a single cell suspension, removing the myeloid percol, using CD11 beta positive magnetic beads, and then removing these microglia, and then we can do gene expression and see, well, what are they making? And so the microglia that have been taken from brains that were exposed to prenatal stress have increased in inflammation. So they make more cytokines, IL-1 beta, TNF alpha, and IL-6 didn't end up being significant. But it does look like there's alterations in cytokine production. And then when we looked at the microglia isolated from CCL2 knockout mice, we no longer see that increase in cytokine production. When we look at the microglia from the antibiotics treated mice, we no longer see that increase in cytokine production. So it looks like that aspect of microglia function is dependent on CCL2 and microbe production. So we think that that's an important mechanism through which microglia might be shaping the developing brain. So prenatal stress, it turns out that the call was coming from inside the house. I don't know if you guys like horror movies. I do not, but a key theme in several of them seems to be like the call's coming from inside the house. So I thought it would be coming from the mother, again, I'll tell you the faucet and the sink, but it really seems like the fetus is, that the tail is wagging the dog. I don't know if everyone's familiar with that expression, but that's how I've been thinking of it. But what about viral infection? And I'm not going to launch into a whole other thing. I just did have the flu with my last pregnancy, and I mentioned to you there's an increased risk of schizophrenia. So I had Adrian do a flu model because I was a live influenza model because I was curious. And what she found was actually the antithesis of what we see in stress. So here she saw a significant increase in the spleen, which is evident of inflammation, a robust increase in IL-6, TNF alpha, and IFN gamma in the maternal, in the circulating cytokines, so maternal serum. And she came out with this work last year before she left and started her own lab. But there is not an increase in neuroinflammation. So this is the inflammatory response gene, antiviral response gene, just different genes that we've grouped according to the labels here. And there was the opposite of stress. So the moms showed a robust inflammation, and the fetal brains did not show robust inflammation. So now my current hypothesis is much more Zen. It's much more yin and yang. I guess it's much more DOHAD, is what Helen would say, as if she was on this call, which is the Parker hypothesis. So, you know, there's really something, a give and take happening between the mom and the fetus, developing fetus, at least as it relates to inflammation. So in summary, I showed you today that prenatal stress influences vertical transmission of microbes, the immune system, the maternal immune system and the developing immune system. I didn't have a chance to talk today about my favorite, tryptophan, but I will tell you that it's also altering tryptophan levels, tryptophan transport, and that's an area of active investigation in the lab. Implications in future directions. I'm very interested in modifying the microbes. I already mentioned we're adding back Paracetterella. There's a few other microbes we're very interested in. I'm not so sure about targeted anti-inflammatory treatments as I once was, but I really do think there's an important critical window here where we could benefit both the mom and the developing organism. But absolutely increase mental health awareness and access to treatment all day, every day. We do not support women. We do not support families. We especially don't support children. We aren't doing enough in this critical time period. at all. And I really couldn't end this talk without saying that risk does not equal inevitability. As I mentioned, I had the flu with my last pregnancy. It's so complex. There's genetics, epigenetics, the environment, your lifestyle. There's so much we can do to modify risks and to just support women, support people, support infants, support growing families. And that's really what I will say over and over and over again until I'm blue in the face. And I also do want to end on a happy note. So these are my 4 kids. That actually should be the fun fact about me, is that I somehow have 4 kids, which is an exorbitant number of children to have on the tenure track. And so this tall one here, he's 5'10" now, I think, 5'11". I have a very tall husband. I had him during my 4th year of medical school. As Dr. Leah mentioned, I was in an 8-year program. So combined MSTP stands for Medical Scientist Training Program. And so I had him during my 4th year. This is Matan, my second born. I had him during my 3rd year of residency, which was a stressful time. This is Talia. She is the ginger you saw in the other picture. I had on my 2nd year, my tenure track, and this is the youngest. I had the flu with her. I also had like a dental abscess. It was a mess. And she, other than being incredibly determined, seems to have had no negative sequelae of that. So I show you this picture in case you're worried about having kids and what would be a good time. There's no good time, but you just, if you want, you can do it if you want to do it. And just look how happy and well adjusted they are, I guess, is sort of the other part of that. And I have to thank my lab. Helen is remarkable. I'll be very sad and happy when she graduates and goes back to, she's an MD PhD student and she will go back to medical school. I wanted her to be a psychiatrist, but I believe she'll be an OBGYN. Teresa Arjasikara led the clinical work that I didn't show today, but she's a very talented public health student. Jeff Galley is my microbe guru, really a wonderful person and really tough statistician. Felix Yang just joined my lab. He's another MD PhD student. Mike Bailey is the mentor I found through Google, best mentor I've ever had. Really just very generous with his time and his knowledge. And then Adrienne Antonsen, a very talented postdoc who's now started her own lab at the University of Illinois. Looking forward to the great science that will come from her and my funding. And with that, I will can stop sharing or I'll just keep sharing, and I'm happy to answer any questions. I'm sorry that we only have 7 minutes left. I explained more as I was going than I had during my practice. So
No,
thank you
that
all
was
for your paying attention.
fantastic. Yes, thank you everyone for giving the little hand clapping emoji.
Oh yeah, I love the
emojis. Now in our virtual world, we need the emojis.
We do need the emojis.
So thank you very much. Ooh, a thumbs up too. That's great.
Ooh,
thank you. No, thank you. That was amazing. And I the explanations are always great, even for us senior investigators, because sometimes we're like, oh, right, I didn't know that. There was a couple of things I was like, really? Especially, learning your favorite hormones, I think is really your favorite metabolites from the gut. That was really great. So please either put your hand up or put a question in the chat, or if you feel comfortable, please do unmute yourself and ask a question. Otherwise, you're going to have to listen to me ask questions.
I want to ask myself questions.
I know, that's right. You'll be the hardest on yourself.
What happened to the...
No, I love that. Oh, Annie. Oh, darn it. I had one all lined up ready to go, but Annie, go ahead, please.
Yeah, lovely talk. So much fun. Lots of cool data. I just had like one interesting thought about your different brain regions for your microglia changes. Do you have ideas about why some brain regions are impacted and why others are not?
Can I add to that question?
Yes, please.
I think you also showed, because of course I paid attention to the sex, right? It looked like it was
you're gonna get it. I knew. Yes. Go ahead. I don't have enough sex differences. I'm very concerned about it. But yes, go ahead. What's your question?
It looked like there were sex differences too. So that's all I was.
There were and there weren't. So full disclosure, my parents study schizophrenia. They study sex differences, and I always said I was never going to study schizophrenia or sex differences because it just seemed gauche to study the same.
I know. I've cited your parents more than once.
Yeah. So I was annoyed. So I think the placenta sex differences, which I didn't show today, are probably interesting. And there is some convincing work from Tracy Bale and others showing that the placenta really does mediate some of the sex differences in terms of differential epigenetic changes and things of that nature. Tracy Bale's model is, and I've had this conversation with Tracy Bale more times than I care to think about, that we have very different models, both in the type of stress that we use as well as the time period. So she's embryonic day 1 through 7, which would make sense. That would be a high time of epigenetic changes. Whereas by the time I'm doing it, the placenta is like percolating through, microglia are going through. And so I have problems with sex differences. I have problems.
The question. Andy's question is the most important one. I was just
Okay.
like, oh, that's so interesting. But I feel bad now because I hijacked.
No. So sex differences every day like make me scratch my head. So the deficits in social behaviors we actually found in both sexes, both in females and in males. And actually this, Lisa and I were talking before. I'm very excited. I'm going to be able to add to my funding line on my slides soon, and it's all about fetal microglia. The grant's all about fetal microglia and how does stress change? Does it change where they come from? Does it change their, there's actually lots of different fetal microglia, so they come from all different places, and where they come from matters in terms of their function. So I wonder if that's a difference. I think there's sex differences in that, in terms of where the sex differences have multiple opportunities to sort of insert themselves, but I'm sort of dissatisfied in that I'm not seeing it in the behavior. So maybe I'm thinking about it backwards, but I sort of look at the behavior and then try to think backwards as to what's causing it. So the fact that there's no sex differences in the behavior has been not
we'll
a
have
problem.
to talk about that later. But Annie, your question was
Annie's question, brain regions.
Right, brain regions.
I think that, so for example, the motor cortex, didn't have a change at all. So we were using that as a negative control. I think that the prefrontal cortex is really important in social behaviors. A version of the grant that didn't get funded was to work with Katie Lenz, and she's very interested in sex differences and microglia, and we really wanted to trace the serotonergic fibers as they went from the into the prefrontal cortex. So I think that could be really important. I think it might depend on microglia and the way they're pruning or the way they're phagocytizing certain synapses, which could change literally the direction that they're migrating through the developing brain. And so I think that the brain regions might be impacted that way. That's one hypothesis that I have that I won't get to test because that version of the grant didn't get funded, but we'll find money other ways to test it because I'm still pretty sure it's right. So that could be one way that's brain regions are some are affected and some aren't. I don't know if that answers your question. There's other ways. I mean, I could come up with ideas all day. I'm not short on ideas. I'm short on explanations sometimes, but.
Wow. I mean, I think we're probably all short on explanations. I'm going to ask you maybe it's a weird question unless somebody else has one.
I see.
Oh, Jesse has one.
Oh, Dan's receiving vagotomy. I hate vagotomies. So I didn't mean to answer your question that way. So what Jesse is very intelligently asking about is the vagus nerve is thought to be very important in transmitting. It's actually the one nerve that sort of is dangling all the way down. And I have to always think back to my medical school education, which is decades ago now, but it's the one that really migrates down and through and all the way down. And is probably the closest other than the enteric nervous system to the gut. And there's a really every field has its controversies. Vagotomy is one in mine, where sometimes if you do a vagotomy, then you get rid of the vagus nerve, then you get rid of the changes in behavior, which would prove that the vagus nerve is what's transmitting changes in the periphery to the central nervous system. But other studies, when they do a vagotomy, there is no, it doesn't do anything. So now everyone's there's lots of controversies in my field. The other is whether or not placentas have living microbes in them or not. And I'm tired of that controversy, but that's a big controversy. So I have not bothered to do vagotomies in my model, but it's possible that it's mediating. It's possible that it's not. Operating on the mice during pregnancy is somewhat of already a little bit of a stressor. So I've just been reluctant to vagotomize that, but it's not a bad idea. It's just already controversial and I'm tired of controversies.
You're tired of controversy. I'm
well,
sorry.
no, I mean,
so much controversy. We're like
the evidence was already
right. It sounds
messy,
like it's
so I didn't know if it would contribute anything other than more mess.
Right. It doesn't sound like it's like the definitive experiment. I always, you know, Jesse, here's one for all of you too. You know, there was something that came out in my area of neurogenesis saying, and this is the definitive article showing proving this. And, you know, it's, of course, it's not definitive, like nothing's definitive. That's why, you know, we'll still have a job 20 years.
Yeah.
50 years from now. That's what I'll say.
No.
What about like, oh shoot, we're 1 minute over.
I know.
All right, well, never mind about my question.
Well, you can email me. I'm happy. We'll meet like we promised.
Yes, we will. We'll definitely meet. And I just want to ask you all to join me in thanking Dr. Gur again for an amazing talk, and thank you for your explanations. And we'll have to get you back to talk about like some mentor stuff. I think that would be great.
I'm all for it. I'm all
I
for it. I love that.
Yeah, I think that would be really.
Thank you guys so much. Thank you so much for the invitation. It's so good to see you.
Thank you. Thank you for coming. And
Everyone stay well. Thank you.